So that's a great piece of work. She's gone on to get her PhD. She's a postdoc now. She's going to tell you a little bit about that. I've been lucky enough to be on a couple of cruises with her. And so, make good friends with your colleagues because you get seeing them over and over again. Thanks, Sacha.
Absolutely. Oh, thank you for the introduction, Colin. Oh my gosh. I haven't even started talking yet. Thank you. It's so exciting for me to be here. I'm really happy to see you all. I also remember what it feels like on the Friday of the first week of the optics class. So I was reminiscing yesterday on my drive up here that I think on this day when I took the class, I went through the whole day feeling like I was on the verge of tears because I was so tired. So if that's you today, it's okay. We're going to go through it together. I hope this will be a pretty fun lecture. I have built in some things for you all to engage. If you get to a point where you're really losing focus and you need another quick break just to stand up, stretch, move around a little, do what you need to do. I will just be here and we can pause and keep going. But I don't want this to feel tedious. So Colin gave a little bit of an introduction. I'm going to talk about phytoplankton today, but first I wanted to give you a tiny bit of background about me and who I am and how I got here. So I did my PhD at UC Santa Barbara. My dissertation looked at phytoplankton community composition pretty broadly, using a bunch of different in-water methods. I'll talk about many of them today, and also some remote sensing methods, which I'm not going to talk about today. I'll leave that for Allie Chase later in the month. And I'm currently a postdoc at Mbari in Moss Landing, California. My postdoc is looking at phytoplankton carbon export in sinking particles. I'm also not going to talk about that today because it's less directly relevant to optics, but if you're interested in that, definitely come find me and we can chat about it later. So as Colin said, I went to Bowdoin, so this is the recent history, but I also wanted to put up a bit of Coastal Studies Center relevant history. So the picture on the left is from my first time ever at the Coastal Studies Center, my first week of college, my first ever oceanography lab. And that was, I just remember coming out on the dock. It looked really different back in those days. None of these buildings were here. And it was so beautiful. And I think it was in that moment that I was like, oh, I can do this as my major in college. And I get to come out here and see this beautiful greenish water all the time for credit. So now you're all getting to experience the thing that really pulled me into this field, in addition to the wonderful classes I took at Bowdoin. And this picture right here on the right is from a really fun day that I spent on the dock down there, where Colin tasked me and my fellow summer research friend, Skylar, who is now living in Santa Cruz with me, to spend a full day on the dock, collect data every hour. She let us go out by ourselves with the instruments, spend the whole day alone. When challenges came up, we had to figure it out ourselves. When our samples started to get warm, we had to drive to the little country store and buy blocks of ice and break them into a million pieces and pack them around our bottles. But I feel a lot of affinity for this dock and this place after spending those 12 hours, some of which with black flies and mosquitoes, as you may be experiencing a bit also. So that's me. And now we'll talk about phytoplankton taxonomy and in-water methods. Putting up Meg's roadmap slide again, just so we're all on the same page. You've made it to Friday. We're here, we're talking about phytoplankton. This afternoon, you'll get to do some work on fluorescence. Ivona will talk more about that. But for now, we're gonna just dive right into the big major topic. So my first part of this is a question that I'm going to put to all of you. Can you define phytoplankton? I saw one head shake already really quickly. It doesn't have to be a perfect definition. I'm curious to know how you would define this term if you were asked to do so. And there's not a wrong answer. The point is that there may be no right answer. Oh, okay. All right. Let's see. Okay, great. All right. Photosynthetic organisms. So that tells us something about how they're getting their energy, photosynthetic, and organisms, their lives. Does anyone else want to give us one other definition you're comfortable with photosynthetic organisms? Okay. Right, exactly. Okay, very small. Something about the size, that's important too. Can't move by themselves. And already you're saying, no, this, I totally understand what you're saying. They're at the whims of the ocean currents, right? But the reaction that you just had to already say there's some complexity there is very relevant to, I think, how all of you are feeling. Were you going to say something else? Okay, perfect. All right, so great. At the most basic, when you first learn about phytoplankton, you may have learned something about the etymology of the word from Greek phyto, so light, that's telling us photosynthetic, something about how they're interacting with their environment, and planktos, to wander or drift, so they are not able to swim against the currents, they're drifters or wanderers. And if you looked at this definition in something like an intro oceanography textbook, you probably would get this very basic definition. They're microscopic photosynthesizers. They're found in the sunlit global ocean, something like that. So very much what you all were able to come up with, something about the size, something about how they're getting their energy. But as you all are probably suspecting, or maybe know already, the more you know about phytoplankton, the trickier it gets to really distill it down to one simple definition. And I think even when you at face value take that first, okay, microscopic photosynthesizers, you all seem to know more than that already. So when I ask you this question, can you define phytoplankton? You kind of froze, and I understand that. So let's talk through why it's so tricky to define phytoplankton. I'm going to start with the tree of life. So phytoplankton have really high phylogenetic diversity. Their phylogeny is talking about where they're placed relative to other living organisms. And so the tree on the left is showing broadly all of the eukaryotic taxa, but we also have bacteria and archaea. And there are a lot of phytoplankton that are members of the bacteria, so this broad kingdom of life. There are also archaea pretty much everywhere in the ocean. So sometimes they might be grouped in with phytoplankton in that they are also too small to swim against ocean currents. And as we develop better methods for sensing them, we're detecting them in more and more places. And so then within the eukaryotic community, that's, organisms, cells with a nucleus like us, we have all kinds of diversity. And this figure on the right is highlighting some of the places where we see major phytoplankton groups. Thank you, Colin. So we're seeing all these major phytoplankton groups that you may have heard of before, but you can see that they're distributed super widely across this eukaryotic tree of life. So even when you try to distill it down to something a little bit more specific, it's not just the bacteria, we're looking at the eukarya now, you still can't get more specific. Phytoplankton are everywhere. I wanted to show a version of this eukaryotic tree of life that also puts us on the map. So if you see this little UR here in the bottom corner right there, that's under the bilatera. So there are all kinds of organisms that have this bilateral symmetry. Basically, if you draw a line down the body, it's symmetrical across for the most part, not perfectly, but that includes us, and it includes a lot of other organisms too. So then when you look at one of these major groups that includes a ton of different phytoplankton taxa, it starts to give you a sense of the fact if bilatera includes us and dogs and cats and all these other things, then where are all of the phytoplankton that are in, say, one of these little branches that are coming off over here, just like how we and so many other things are encompassed under that one branch of the tree. So that brings us to this next challenge. Phytoplankton have really high taxonomic diversity. So phylogeny was describing kind of where they are in the tree of life. Taxonomy is then how we're defining that within that tree. So we've moved down from the kingdom. We're looking, if you remember probably from maybe a high school biology class or earlier, there are all these little strategies we can use to remember kingdom, phylum, class, order. And so now we're looking down at getting even more specific with our taxonomy for phytoplankton. This is showing within the eukarya some of the class levels where we find phytoplankton. So there's the Bacillariophyceae, those are all the diatoms. That's a huge taxonomic group, but it's just represented here as this one class. And the phytoplankton are found throughout all of these different branches, as we've already talked about. So over 25,000 distinct phytoplankton species have been identified, but that number is definitely only going to go up. And there's so much of the ocean that we have not been able to explore yet. So the number of phytoplankton we could possibly find is also going to continue to increase. All right, so we have the phylogenetic diversity, we have the taxonomic diversity. Within all of that diversity, there's also all of this morphological diversity. So that's referring to the shapes and sizes of different phytoplankton cells. So this figure is taking that tree of life that we just saw, showing all the different branches where we find phytoplankton taxa, and then you can see this huge variety of cellular shapes and sizes that we're getting across all of that taxonomic diversity. Ivana had a really nice figure that she put together, so I borrowed it from one of her old lectures, just talking about putting this into context the scale of phytoplankton size diversity when we think about it in terms of our own size on Earth. So you may have heard people talk about these broad categories of phytoplankton sizes, these major size classes. There's the picophytoplankton, which are between about 0.2 microns to 2 micron diameter, then the nanophytoplankton from 2 to 20, and then the micro, which are greater than 20, giving maybe an upper limit around 200 microns, but some can be a little bit bigger than that. And so as the pictures show, if we're thinking about that scaling in terms of our own bodies compared to other organisms on Earth, or even just structures on Earth, when we get larger than the organisms that we know, we're going from people to whales to the Statue of Liberty, and those are all phytoplankton in that micro scale. So you can see that it might be a little bit difficult with all of these complexities we've talked about so far, but now let's dig into that one that came up as soon as we started talking about the definition of phytoplankton, that they're photosynthetic. Not always. Unfortunately, phytoplankton, well, maybe fortunately for them in terms of their life strategies, but they have a lot of different ways of feeding and getting nutrition. And so organisms that we refer to as phytoplankton can be phototrophic, which is a common strategy for phytoplankton. We talk about that a lot, particularly in optics with their interactions with light, but they also can be heterotrophs. They can be mixotrophs, which means they perform both phototrophy and heterotrophy. And there are also parasites that are classified as phytoplankton, or phytoplankton that can have parasitic life stages. So we can't even call phytoplankton photosynthesizers and really safely have that as a catch-all unless we want to obscure some of that complexity, which often is useful for the case of explaining it to non-experts. I just Googled mixotrophy to try to see what type of visualizations we could get and found this incredibly cool picture of a carlidinium, so it's a dinoflagellate. It contains chloroplasts. We can see the green under the microscope. We know that it is capable of being photosynthetic, but it is also consuming this other organism into its cell. So this is an example of what mixotrophy would look like in the environment. And you can see how that would make it really complicated to try to describe how this phytoplankton is existing relative to some of these categories that we might want to put it into: its size, its shape, how it's getting its energy. You can see even just from these three images, the size and shape of this cell changes a lot, and the way that it's getting its energy is actively changing as it goes through this process. So I've now posed a lot of complexity and exciting variability into the phytoplankton definition. I'm guessing many of you have in some way heard about describing the composition of a community of phytoplankton. You even got to practice doing it yourselves a little bit the other day. So when you hear that, you describe the community composition of phytoplankton, what are some of the ways that we can define that composition? What are some of the categories that we put phytoplankton into? Yep, taxa, so taxonomy, that's an easy one. Size, exactly, right. So those two automatically, taxonomy, size. Luke, I saw you raising your hand. Okay, size, taxonomy, yeah. Functional types, absolutely. So what's their function in the environment, and how does that relate to the rest of the Earth system? Is there anything else? I think you've... You're heckling. Okay, perfect. We need some heckling. Mix it up. There was one more that I thought of, but if you don't think of it, that's okay. There also might be one that I didn't think of. What are maybe some traits that phytoplankton possess that might be relevant to thinking about other processes on Earth? It's sort of related to functional type. Like maybe nutrient cycling. Okay, all right, we're gonna go on to the next slide. So pretty much everything here right away: taxonomy, particle size, function, role in the environment. And so the other one that I put was chemistry, because phytoplankton can contain either pigments or minerals that we might be interested in tracking, or just elements in the case of maybe silica, right? Or calcium carbonate, things that we are interested in tracing throughout the ocean and that are in phytoplankton cells. And so when we think about defining community composition in these different ways, these different ways of describing and separating groups of phytoplankton can be relevant depending on your goal. So if you are interested in targeting a phytoplankton community using remote sensing, some of these ways of describing phytoplankton might be more relevant than others. Or if you're interested in building an Earth system model and you have a certain number of phytoplankton groups, it might be important to include phytoplankton that have a particular function in the environment or a broad variety of sizes and shapes. So a big part to me of thinking about defining community composition is thinking about your question and then thinking about how you can answer your question by describing your phytoplankton community as accurately as possible within the bounds of some of these categories, which, as we've described, can already be really tricky, and there can be these kind of blurred boundaries. All right, so I'm going to walk through some major phytoplankton groups that you might kind of generally hear about. If you stay in oceanography, even if you're feeling like you're maybe someone who's not a biologist, it's good to know some of these general phytoplankton groups. I'm just going to give a little background. You definitely don't have to remember it, and you can always come back and reference these slides if you want a really quick reference. So we have Synechococcus to start out. This is a super abundant cyanobacteria that's really small. And when we think about ways to separate different phytoplankton groups, the big one for Synechococcus is that it has a phytoplankton pigment that fluoresces in a unique wavelength. So you can use that phycoerythrin fluorescence. Ivana will talk more about fluorescence in her talk next, but you can use that to separate these tiny, tiny cells that can be hyperabundant, especially in some of the warmer, kind of more tropical areas of the ocean, and you can separate them from other phytoplankton cells. The kind of sister to Synechococcus is Prochlorococcus, which is also cyanobacteria. This is the smallest known thing that we describe as a phytoplankton. So it's about 0.7 microns in its cell size. That can vary a little bit. There's some interesting observations of that cell size varying throughout the cycle of the day as these cells grow and then divide. You can see this really cyclic pattern of their cell size, but generally they're really tiny, and they uniquely contain two accessory pigments that set them apart from other phytoplankton. So when we think about, again, what makes these phytoplankton different from all other phytoplankton, for Prochlorococcus, their pigments are an easy way to separate them. The last member of our cyanobacteria family for this broad overview is Trichodesmium. So Trichodesmium are also really small, but they form these kind of clumps or tufts in the ocean that can even be visible from space. In terms of their functional role in the environment, they're nitrogen fixers, so they have a unique place in that nutrient cycle. So when we're thinking about these different ways that we can divide things into different groups, this one has an interesting functional role. It potentially has a cool remote sensing signal. And like Synechococcus, it also contains this unique fluorescent pigment, so phycoerythrin, that will fluoresce at a different wavelength. I wanted to just, since we are talking a lot about remote sensing in this course and optics, put up this quick image of what it looks like to see a Trichodesmium bloom from space. So you can see these filaments that are really bright white in contrast to the ocean water around them. And so there have been attempts to develop algorithms to detect Trichodesmium from space. And because it looks so different from a lot of other phytoplankton groups and from this kind of background water signal, that's been pretty successful in areas where they are in these high abundance. And you can see this really clear signal, both in a true color image and in the reflectance spectrum. All right, so that was our cyanobacteria side. We're going to move into the eukaryotic side of things, which is lots of diversity, as we already talked about. A big one right away is diatoms. So diatoms are members of the red algal group, which is referring to the types of pigments that they have. There's a huge range of sizes for diatom cells. So I want to highlight that because we often talk about diatoms as being members of the microphytoplankton group, so that biggest phytoplankton size class. But there are diatom cells that range from around 3 microns all the way up to 200 microns and maybe even larger sometimes in diameter. So if we're thinking about diatoms as only members of one size class, we're missing all the diatoms that exist in that 2 to 20 micron size range in the nanophytoplankton. So again, thinking about classifying things into these different broad groups, it's important to not be too prescriptive and miss some of the natural variability in that community that makes it really interesting to look at all of these different phytoplankton groups in their environment. Also, in terms of their role with nutrient cycling or the minerals in the environment, their cell walls are made of silicon, which contributes to them being efficient carbon exporters because these dense silica walls can help diatom cells to sink more efficiently. And so again, thinking about what is your purpose for defining phytoplankton community composition? If you're interested in building a carbon export model and you want to know something about the groups that are contributing to carbon export, you might be really interested in classifying the types and amount of diatoms that you have in the ocean if you think they're going to be a big part of your carbon export story. Moving on to the dinoflagellates. Dinoflagellates also have massive taxonomic diversity. They can also get a little complicated for a lot of these different in situ methods. So I'll talk about that a bit more specifically later, but some of the important complications you can know going into it, like the diatoms, they have a really big size range. Some models or classifications will also put the dinoflagellates only in that micro size class, but there are a lot of nano-sized dinoflagellates as well. And so something that can make it tricky to detect dinoflagellates in the environment is that many of them are mixotrophic or heterotrophic. So their feeding strategy is not always purely photosynthetic. And that often means that the amount or types of accessory pigments that they have, or even the amount of chlorophyll A per cell that they have, can be really different compared to the relationships that we see in other phytoplankton groups that are only performing photosynthesis. So if you're using a method that's depending on being able to detect phytoplankton pigments and hoping that you can characterize your dinoflagellate community, you might miss the fraction of the dinoflagellates that are not using photosynthetic pigments to get energy, that are heterotrophic, that are acting more like microzooplankton than like phytoplankton. Also, dinoflagellates often come up when we talk about harmful algal blooms. They're not the only group that can form harmful blooms, but they are often part of these red tide species. So a lot of times when you hear about these big blooms happening, the culprit might be dinoflagellates, depending on your region and your time of year. I wanted to show a quick example of what that can look like. So this is an image from National Geographic of a massive dinoflagellate bloom off the coast of Florida. Has anybody heard about the type of dinoflagellate that causes these harmful blooms? Yeah, exactly. Right. So this is Karenia brevis, and you can see, especially when we're thinking about doing this from space or doing this from above the ocean and looking at the color of the ocean, you can really clearly detect from this image where the Karenia are found compared to the ocean water that probably still has a lot of cells, but in much lower concentrations than in these areas where it's really concentrated. And so being able to determine when a harmful bloom is happening, which members are making up that harmful species is really important, but we also sometimes need to dig a tiny bit deeper, because sometimes when dinoflagellates are present, even if they can form harmful blooms, they're not always performing that harmful function. So you can have these ways of either with remote sensing or with an observatory that you're not actively visiting, detecting that those groups are in the water. And then you can maybe go test and see, are there toxins being produced? What is the actual effect for human health? Do we need to have more action happen to protect people and fisheries and maybe even animals in the region? But something like this with a dinoflagellate bloom can be really significant, as you can see from this image. All right, so we're staying in the red algae for just a little bit longer. The promesiophytes are another group with a huge amount of taxonomic diversity, and I'm going to talk mainly today about two really different groups. So we have calcifying phytoplankton that includes coccolithophores, you've probably heard of those, which are really highly scattering. So they have these calcium carbonate shells around their cell that are going to scatter a lot more of the light than a typical phytoplankton that doesn't have these kind of white scales coating its cell. So that gives us a unique opportunity to think about detecting it based on the minerals that are in the phytoplankton, its actual body. Also, another one that comes up a lot with the promesiophytes is phaeocystis. So phaeocystis, in terms of thinking about function in the environment, is producing dimethyl sulfide, and it can form these big clumps that often can then, from this gas production, lead to foam. So you might see on the beach, this happens a lot in the UK, there will be these big clumps of foam. I think I put a picture in a couple of slides, but this is also something that we're concerned about in terms of human health. There can also be a lot of bacteria cut up in that foam, so it can make swimming at the beach a little bit more hazardous for people if it's near an area where bacteria is aggregating, like an outflow from some wastewater or something like that. So between these two groups, which I'm showing, here's the blown up picture of the coccolithophore, but phaeocystis is down here on the bottom, and you'll see that a little bit bigger in a second. Those two look completely different from each other. Their function in the environment is totally separate, but they're both members of the same taxonomic class. So again, thinking about how we're describing our phytoplankton community, if you're most interested in knowing more how they're behaving in a remote sensing image, or how they might be affecting human health and well-being for people who live along the beach, it might not be enough to just say, we have promesiophytes in the water. Yeah. Well, so they evolved similarly. So in terms of the tree of life and their placement, they are similar taxonomically because they evolved to have similar pigments. They have similar life histories that got them to this point, but they are really, really diverse. So let me quickly go back and see if we can look at that. Here we go. So if we this whole group of the Hecrobia right here, this entire broad taxonomic group includes the promesiophytes. Here it's highlighting the Coccolithophorids, which is going to be, I guess, let's say like a suborder of the Promesiophyceae. But you also have cryptophytes, which are completely different functionally, are in that broad community of the Hecrobia as well. So it all has to do with how they evolved, how similar they are to each other on a genetic level, and not necessarily then if that reflects their function in the environment. So right, in terms of classifying phytoplankton, I think that type of kind of historical biological taxonomy, you need to understand that part of it because it fits them into this kingdom of life, these historical observations, these records that other people have from things like microscopy or other observations throughout history. Yes, of course. Yeah, please.
Sides. I'm very sorry.
No, go for it.
Pretend like this is not an interruption. Sorry,
okay. So
it's
it's
recording wrong.
Frustrating because you have to understand that, but then you also have to know that what you're talking about is going to be really different.
Yeah. I think of an analogy when we're talking about how similar the different taxonomies are.
Yeah.
Like, is there an analogy we made with like how we think of animals, like when we comparing humans and apes, comparing humans and all other mammals, humans and reptiles, like how?
Good question.
Is there an analogy?
Yeah, well, so I guess maybe it'll be helpful to look back at this version of this eukaryotic tree of life, right? So in some cases, comparing phytoplankton to phytoplankton can be more like humans and apes. So it's things that followed a similar evolutionary path, and then at some point their evolution diverged, and they're similar, but they're different enough that they're totally different species, and they interact with their environment really differently, right? So in some cases, that's a great analogy. In other cases, it's maybe more like humans and dogs. So you are both mammals, you're both bilatera, right? You both have certain ways that you interact with your environment, but you would never, ever be considered the same species. You look completely different. The way that you move is totally different. And then because this is, part of this crazy, really high phylogenetic and taxonomic diversity is that in other cases, phytoplankton groups are as different from one another as we are from phytoplankton in terms of this type of taxonomy. So if we're over here with the bilatera, and then let's see, the diatoms are right over here. But so diatoms are a branch that's comparable to where the bilatera are over here. And diatoms and bolitophytes, those are both phytoplankton groups. They have similar pigments, but they're totally different branches on the class level. So they are really, really different in terms of their taxonomy, their evolution, their similarity to each other. So it's difficult to come up with a good analogy. Yeah. Is there what? Another way across, not that I know of within this type of traditional taxonomy. So I think something that's really cool about oceanography and marine science is that you can't do it in a vacuum, right? You have all these different disciplines that are coming together. And so taxonomy is something that was designed not for marine science application necessarily, but that we use these tools and we kind of fit our organisms in with this tree of life. And there are biologists who are working in marine systems, and there are marine scientists who are using biology as a tool. And so to make kind of a better classification, you could probably have a more specific description of just phytoplankton groups. But in terms of placing them within this broader taxonomy, there wouldn't really be a better way to do this, because if you're placing them on this phylogenetic tree, you're using DNA as the way to do that. And so that's something that, goes beyond just classifying phytoplankton. That's for classifying all other organisms too. Yeah. Ivana, yeah.
Think about, like, evolutionary wise, when did they reach, there's still a world, but, how do you know to find them, look at them, way before you. So you have to do a little bit of evolution and putting in your mix and what you know. So this is insane, that's what it makes it all amazing, side of them, because if you're not using this conventional biology, what else do you use? That's one of the important things about suffering, explaining something. I will explain to you what I did, so you can go and feel it and experience it. So kind of really, it's insane. But here's the text about it.
Yeah, no, and I think, yeah, you brought up a really good point also, Ivana, about that, the fact that phytoplankton are unicellular. And so both the amount of time that they've had to evolve and the fact that they have these shorter generation times being only one cell, anything that's multicellular is going to take longer to evolve because it's harder to divide and grow, right? So phytoplankton have been on Earth for a long time. All these different types of cyanobacteria have been around for, billions of years, right? And so if you have had that long to evolve and change and you're dividing and changing constantly, you have all these other ways that this taxonomy has branched off and that things have differentiated from each other compared to maybe higher order mammals. Yeah, it's, when I was applying to grad school, I think I said to Colin at one point, I don't want to do molecular work. I'm not interested in understanding phytoplankton on the DNA level or knowing about evolution or any of this. It's a little bit inevitable. If you're going to understand phytoplankton community composition and taxonomy, you need to understand the evolutionary history a little bit as well, because it's all connected. We'll get into it a tiny bit more later, but stay tuned for that. I think that'll be fun. Okay, we're going to go from boring slide with facts to fun slide with photos. So thinking about detecting different phytoplankton taxonomic groups from space, another one that we can do pretty well is coccolithophores. So because they have those highly scattering calcium carbonate shells, that means that they can almost look like kind of milk stirred into the ocean. They make these beautiful, beautiful blooms when they're forming often at high latitudes. So you see Norway right there, where, there's a limited growing season for phytoplankton. And then when they grow, they can form these really gorgeous blooms that you can see from space in true color images, or also their signal and reflectance. So you were all thinking a lot for your lab today about scattering, right? And so it's that scattering signal from coccolithophores that makes them unique from a lot of other phytoplankton. So thinking about those ways that we can have something we can define in the biology world, the taxonomy, the coccolithophores, and then something we can define in the optics world, having these highly scattering cells, putting those together to say, okay, we can detect this group using this technique. And a beautiful example happens right there. And then I promised that I would show that foam. So here's the Phaeocystis foam that can form. This image is just showing what it looks like when this whole kind of colony forms in one beautiful, perfectly round ball. But often you'll see these different cells. You can see it's a bunch of individual cells in this polysaccharide gel matrix. And I know I saw over on the IFCB activity that you found some Phaeocystis in part of your time series. So that's pretty cool. All right, so the dictyocophytes, I included this group because when we think about salicifying phytoplankton, we often only talk about diatoms, but there are other things that are incorporating silicon into their cells in the ocean. So thinking functionally and about nutrient cycling, when we're thinking about silicon in the ocean, we also need to think about the dictyocophytes. They're generally nano-sized phytoplankton, and they also have these really beautiful looking cells with this silica structure that you can see really nicely in a microscope or in another type of instrument that can take these types of images of phytoplankton cells. All right, rounding out the red algae, we have the cryptophytes. These are pretty small. They're in the nano, but often on the smaller end of the nano-sized phytoplankton. I already gave that example of how they're kind of broadly grouped into the Hecrobia, so with the promesiophytes, even though they're really different. And then the cryptophytes, Fortunately, we have a fun way to detect them as well. They uniquely contain the pigment alizanthan. So we can use that as an indicator of when we might have cryptophytes in the environment. Although since they're often small cells and sometimes they're not in super high abundance, it isn't always a great way to be able to separate them from other phytoplankton. Sometimes it just means that they're co-occurring with other groups. All right, so you all have been thinking a lot about one member of the green algae, everything that you've done with Denaliella the past few days. That is our kind of token green algal group. The chlorophytes also contain crazy taxonomic and morphological diversity. And I'm going to show a slide in 1 second that will explain that a little bit, but just to quickly let you know, they are mostly in the nano size range, although they can really span that whole nano sized range. So they can go from the smallest end to the largest end, and they contain chlorophyll B. So that's a way to separate them from other phytoplankton. That's their major accessory pigment. We'll talk a little bit more later about some ways to detect chlorophyll B in the environment and how that can be used to separate chlorophytes from other phytoplankton. So when we're talking about phytoplankton evolution, I thought that this illustration of the chlorophytes, the green algae specifically, was really useful because we're going from this, hypothetical, that they're saying, ancestral green flagellate. So we know that originally there was one common ancestor. And again, we know that from using these molecular methods and doing some DNA sequencing, but that branched off into two completely separate groups. So we have all of the land plants on one side, and think about the diversity inherent in saying land plants, right? If you just look out the window, you could probably find 50 different types of land plants. And then on the other side are all the different things that we're describing in the ocean as chlorophytes. So the Denaliella are in the Chlorophyceae group over here, but this doesn't look like a Denaliella cell, right? If any of you looked up what Denaliella looks like when you were doing these labs, those are unicellular. They don't kind of group together like this. They do have flagella, but they're not on both sides of the cell. So even within kind of a subset of this broad group, we know that we're already getting different sizes, we're getting different cell shapes. There are all kinds of things going on. So again, we're left with this challenge. Phytoplankton are really difficult to divide, and we need to try to do our best to do it because it's important to divide them into these different groups and understand their function in the environment, their interaction with the physics and chemistry, their interactions with each other, right? How we're able to detect them using optics, either in the water or from remote sensing. So I want us to try, and I'm going to try to make this a tiny bit more interactive, but I want us to try practice separating phytoplankton into different groups using some set of criteria. So we're going to split into two halves of the room. This kind of half can work together and this half can work together. And I want you to just talk in small groups for a second. Maybe we'll give you like a minute and a half because you don't have to get it perfectly right, but talk about which groups you would put in each of these three categories. And I put these pictures on the bottom, but if you want me to remind you of the names, you can definitely ask. But we have the microphytoplankton, the nanophytoplankton, the picophytoplankton. Where would you put all of these different taxa? And it's okay to put things into two categories or three. Ready, go.
Oh, sorry.
All right, let's go to another 30 seconds. You're doing great. Hearing good conversations. Okay, all right, so stay in these groups because you're going to do this a couple more times in slightly different ways, but how did you all divide up your phytoplankton groups into these different size classes? What did this group put in the microplankton? Diatoms, awesome. Okay, did you guys have that? Diatoms and dinoflagellates, great, okay. Anything else? The primnesiophytes, awesome, cool, okay. How about in the nanoplankton? What did you have there? Okay, diatoms, awesome. Also dinoflagellates, you said? Perfect. How about you guys? What did you have? Coccolithophores, cool, awesome. Anything else? Cool, yep. Lots of things in the nanoplankton. How about the picoplankton? Cyanobacteria, great. Okay, all right, so this is what I put in this category. And I don't know if there are ways that this looks surprising to you. If there are, maybe we should talk about them, but I think you got most of what I was thinking here. The major thing that I think was maybe tricky is that trichodesmium, because they can form these clumps and tufts, can be really different sizes, which is a little bit cheating maybe, but you might find them across different size classes if you have one individual cell compared to if you have a tiny clump or a slightly bigger clump or a really big clump. And there can also be some chlorophytes that are pico-sized, although I don't know that I said that, so I don't think you necessarily should have gotten that. Yeah.
When we're in a clump, you don't look at them as an individual.
Well, so that's a really good question. I was thinking about this in terms of if you're trying to classify something by its broad size class, you often are maybe thinking about like an optics or remote sensing method. So I think it's important to know that you could have things that look bigger in a group than they do on their own. But if you are thinking about it on an individual cellular level, you're right that they would only be in that picoplankton category. But in the environment, you'll probably find them often in these clumps. Yeah. Exactly. Diatoms, right, that form chains. So if you already have something that's a microplankton and then it starts forming a chain, it's only going to be even bigger, but you could have nanophytoplankton that then are forming a chain, and then you'll observe in this kind of more micro size class, right? So again, I think some of that depends on the methods you're using, what you're hoping to describe in the environment, but you'll see things in both of those classes in terms of whether they're in a chain or they're individual cells. Yeah. Okay, now we're talking a bit kind of vaguely about the function. Maybe the box sizes will give it away a tiny bit, but if you can try to divide these phytoplankton groups again into some of their function in the environment, we'll do it pretty quickly. No need to get them all, but let's give that 2 minutes. Go for it. Bless you. Bless you. All right, let's talk about it. This group over here, do you have any category you want to put up on the board? Do you have anything that you felt really confident about? Yes. Yeah. Yeah, so right, so solidifying phytoplankton, diatoms, and dictyocophytes, awesome. This side, anything that you felt really good about? Awesome, yeah, cyanobacteria, dinoflagellates, and diatoms can form harmful algal blooms. Not necessarily the cyanobacteria that we talked about directly, but definitely cyanobacteria, and that is exactly how I also think about it, can form harmful blooms. And you often hear about that in inland waters. They're a really, really big problem. Yeah. Yes, yeah. Right. So I feel like even in the past couple of years in this region, there's been more problems with cyanobacteria and more education, especially for people who walk their dogs, like in the woods, and maybe there's a pool of standing water and a dog drinks from it. It could have one of these harmful cyanobacteria in it. So it's a huge human health hazard. Anybody have an idea for calcifiers? Either group. Cogolithophorae, awesome. How about nitrogen fixers from these ones that we talked about? Trichodesmium, awesome. And then DMSP producers. Basistis, okay, awesome. You guys did great on this one. The only thing that I added, I didn't talk about the chrysophytes because they're a big class in the freshwater, but there's only a couple of different types of chrysophytes that have been found in the ocean so far, but they are also silicifiers. So I just added those into that silicifying phytoplankton group. But otherwise, fantastic job. Nice work, everybody. Yes. Good question. I don't know as much about if it's in estuaries or, I mean, anywhere that there's fresh and saltwater meeting, I'm guessing that you would be more likely to find freshwater taxa. So it's, yeah, probably exactly where you would find them. Cool. Okay. I have one more challenge for you all, but it's a little bit cheating because I haven't talked to you about pigments yet, but I want to know what some of your assumptions are. So you may have heard maybe in interoceanography or in some papers that you've read about fucosanthin, which is described often as the diatom biomarker pigment. So I would love for you to just take another couple of minutes to quickly discuss which groups contain the diatom marker pigment and which ones don't. Just your best guesses. I'm not expecting you to know anything that you haven't learned before. Yeah. All right, let's just talk about this one. Give it your best shot. What besides diatoms do you think contains the diatom biomarker pigment? Okay, I heard all the red ones, which is a great guess and exactly the right way to think about this, right? So we talked about broadly the red algae, they share certain accessory pigments. Pretty much all the red ones contain fucoxanthin. So I'm just going to put this up so we can talk about it. So talked about all these different classes of red algae that were really different from each other, were really different within their different types of cells. And then we think about ways that we're detecting phytoplankton in the environment. If we're using one pigment and saying it's only in one phytoplankton group, we know in the case of this pigment that that's not true. So there are some things that we can separate. Broadly, we can say the red algae are different from the green algae. We can say we don't find fucoxanthin in cyanobacteria, but do we always know that that pigment is always in diatoms all the time? I'm seeing a head shake and I agree. I would say we do not know that that's always the case all the time. So we talked about a lot of the challenges. I'm just going to show some other very quick visualizations of these challenges from my perspective, because we talked about them kind of individually, but we also have intersections. So this is showing size and taxonomy, which we've talked about as I've kind of walked you through all of these different groups. But if we think about the portion of these different taxa that are in each size class, and the divisions here are a little bit different than that kind of standard pico, nano, micro, but broadly you can see this diversity of size. You have a lot of things that are in every size class. You have some things that are distributed really weirdly, where you have a lot of them that are smaller and then some that can be really large. And some of that has to do with, again, forming these clumps or chains. But doesn't seem super straightforward to me to use size as an indicator of phytoplankton when you have all this diversity within each taxonomic group. Then if we bring in one more complicating factor of the feeding strategy, so in this figure, you're kind of moving from the bottom to the top from with the size classes, and then along this x-axis is the different feeding strategies. We have all the protists. So that's one other way that people might describe phytoplankton sometimes, is to call them protists. But protists as a taxonomic descriptor doesn't include bacteria. We know there are a ton of bacteria in the phytoplankton. So again, you're looking at all this taxonomic diversity, all these different ways that they're getting their energy across all these different size classes. And that's not to say anything of the different morphologies that are represented here. So the different sizes and shapes of cells. I mean, this is pretty nuts, right? So now we come to this big challenge. How do we measure phytoplankton community composition? How do we do it? We have so many different types of phytoplankton. We have so many different ways that we can categorize them, and ways that they're obviously different from each other or annoyingly similar to each other. And we know it's really important to be able to separate them on these really broad scales. So I've been talking for about an hour. I would propose we take a 5-minute break. You can think about these questions while you quickly use the bathroom, get some water, whatever you need. And then we'll come back, and after 5 minutes, we will get into it together. Does that sound good? Awesome.
What? So bacteria are included in phytoplankton? Some of them, not all of them. No. So like some, like in the lab that I was working in, like biological geography, they were looking at interaction of bacteria. Are those bacteria considered like ones that are like providing nutrients and are they like phytoplankton cell or like free living? I don't know. I think some people would consider those ones to be other people aren't. Right. So okay, so yes, that's great. But you know, it doesn't look like cyanobacteria, that is a bacteria. We obviously call that one a phytoplankton. But maybe something, you know, some part of the bacteria attaches to a phytoplankton cell or lives like right in the yeah. Okay. Interesting. I think you a little bit maybe just have to define your own definition and then like hold on to that in your head of like, I can put it and I don't see that, and here's why, because I know enough to get the differences. Thank you. Great.
Yeah, that makes sense.
Okay, ready? Yeah. All right, so I don't know, you're good. All right, so did anybody spend time during the last 5 minutes thinking about this, or were you all like, oh my God, give me a break for 5 minutes? Yeah. Yeah. Yeah, yeah. Okay. Yeah. Awesome. Yeah. Okay. Okay. And so what type of tools do you use to measure them, to divide them into those different groups? Okay. That I think honestly, that answer is perfect. The answer depends on the trait. That is a great place for us to start this conversation. So what I was really hoping we could get at in this next part of the talk is, now that we know all these challenges, we have all our important reasons that we want to detect phytoplankton community composition, what are the tools we're going to use to get there? And I want to kind of propose to you all to start this out, that I think a big part of that depends on the question you're trying to answer and choosing the right tools for the job. So exactly what you just said, it depends. We choose our methods that way. So Ivana alluded to this a little bit earlier, but if we look back at the history of observing phytoplankton, that can tell us a little bit about how we got to where we are today, and maybe how some methods have developed out of necessity from the historical methods that we were using. So the first earliest efforts to describe phytoplankton communities, if we think about the fact that they are extremely tiny, one of the words we first came up with when we were defining them, microscopic, you can imagine that for a very long time, people looked at the ocean and didn't know about the diversity of organisms that was there. But the development of tools like microscopes, even if they weren't anywhere near as powerful as the microscopes that we have today, allowed early scientists to go out and take these observations in the global ocean, try to get this massive diversity as much as they could, which, you know, it's crazy to me to think about doing this in modern times. So doing this in the 1800s must have been something to behold, but we could start to get these observations of some of the largest phytoplankton, and specifically phytoplankton that maybe have some of these features that allows them to be preserved or observed more easily. So we're looking here at different types of salicifying phytoplankton, which we've talked about today as this kind of broader functional group, or maybe a way that we can look at nutrient cycling. But it also meant that in these early ways of finding phytoplankton in the ocean, getting those cells under a microscope, these are some of the things that stayed intact as that water was maybe kind of jumbled around, and the microscope had to, you know, move up and down on the boat. But so we have these beautiful observations, and some of these are phytoplankton that we've talked about today, some of the diatoms. Others are not phytoplankton specifically, but relatives of phytoplankton like Rhizaria or Phaeodaria. But these are all things that are forming silicic structures like the diatoms do, and we can get these really beautiful drawings that Ernst Haeckel made. So when we think about these historical observations of phytoplankton and the tools that we were using, our understanding of which phytoplankton are where was really limited by the types of tools that you could use and the places that you could go. So that type of, you know, global ship going all around the oceans, collecting water, using your microscope. That gave some understanding of which phytoplankton are where, but those methods were really limited. And I would argue that today, as much as we've made a huge amount of progress, our observations are still limited, because going out on a boat can only give you so much information, and collecting water samples from one place and time, no matter what type of methods you're using, is going to just give you a snapshot. And so we need to try to think about ways that we can combine methods like remote sensing approaches that are giving us this broader spatial and temporal observation of the Earth with these higher resolution in water methods, where we can get a really beautiful, complete picture of the taxonomy or of the different sizes of phytoplankton that are in the water. So, I'm just putting up here that Synechococcus and Prochlorococcus, two of the major cyanobacteria that we talked about today, those are everywhere in the global ocean. You find them all in all these different places, different latitudes, cross longitudes, at different depths. These things were only discovered, in the last 50 years. So thinking about the fact that that much major, yeah. Okay, I was trying to be generous with that, but apparently I did the opposite. So I'm sorry, Ivona. In the last 46 years, two beautiful organisms have bloomed into the global space, Synechococcus and Ivona, and and Prochlorococcus. But if you think about how, to me, that's really exciting, right? That we have had certain tools for a really long time that limited our view, and then we've developed better tools, and that pace of progress of finding new methods, taking methods from other fields and applying them to marine science. Things that we talk about like HPLC for pigments, or using flow cytometry. A lot of these methods weren't necessarily developed to be applied to the ocean, but when we apply them to the ocean, we have major discoveries about how ocean biology works and how it intersects with all these other parts of life as we know it on Earth. So, the modern corollary to those early observations is to have these large global scale sampling expeditions and maybe use some higher resolution methods. But again, if we look at this ship track, as exceptional and impressive as this is, we're missing huge chunks of the global ocean, right? So again, our task is to try to bring together the high resolution methods and the high resolution sampling in space and time, and somehow find that in between space where we can describe our phytoplankton communities in a way that is important for understanding Earth system models, understanding carbon export, understanding food web structure, but also not obscuring too much of this high variability in different types of phytoplankton that we know exists. All right, so going to talk through a number of different common methods for detecting phytoplankton community composition. And these will each be repeated on their own slide, so don't worry about getting them all down right now. There are obviously some other methods that people are using out there. I'm going to talk about what I consider to be the most common ones, but if you feel like I'm missing a major one that it's important to you that we talk about, please don't hesitate to bring it up, because I think it's good for us all to be on the same page, and there are new methods developing all the time. And I said this quickly at the beginning, but I'm not going to go into the remote sensing methods that we can use, which is its own fascinating and exciting topic, but you'll hear obviously a lot more about that as this month goes on. So again, framing this discussion in this way of thinking about choosing the right tool for the job. We have all kinds of methods. We need to choose either one method, or maybe depending on your budget and your person power and your sampling access, multiple methods. And then we also need to think about, if we're choosing multiple methods, how those methods fit in together and how they compare to each other. Are they giving you complementary information, or are they giving you contrasting information that you need to try and reconcile to arrive at one answer? So when we think about this huge variability in the different sizes and taxa, and then just one axis, thinking about the size and the variability in some of these major instruments that people are using commonly to sample phytoplankton communities, or phytoplankton plus zooplankton, or phytoplankton and other bacteria, we need to think about how the questions that we're asking about our ecosystem then relate to the tools that we're using. All right, so I already talked a tiny bit about light microscopy, but this is, the historical method of looking at phytoplankton that we're finding in the ocean. And it works for a reason, right? So this method has been used for a long time. Microscopes have gotten better and more powerful. We can add to this method now by doing things like staining or dyeing cells. You can change the amount of light that you have under the cells, so you can highlight certain groups or maybe make it easier to see the contrast with other groups by turning some of that light down. But in this method, you're collecting a really small amount of water. Oftentimes, you're kind of concentrating the cells that you have in a larger volume into a smaller volume that you can then look at. And then you're counting and identifying the cells under magnification. So I think this is a really useful method in terms of looking at often the larger size of phytoplankton, because some of it is limited by the user. So as humans, you're looking into a microscope, you're the one who's responsible for identifying and counting these cells. And if you've ever looked at a microscope with all different kinds of phytoplankton, it can be really overwhelming. And you maybe don't know where to start. And so maybe your eyes are drawn first to the bigger things, and then you count the smaller things in the background. But this is a really useful method, especially in terms of thinking about using existing tools if you don't have a huge budget. So if you're in a place that has access to a microscope and you need to know what's in the water, you can get a quick look by just scooping up some water and putting it under your microscope. So a lot of harmful algal bloom species are often bigger. It's a great way to be able to look at those cells if you need a quick answer about what's in the water. And you also can compare it really easily to this historical record. So let's say you have a coastal time series and you've been collecting water samples for the last 80 years and looking at them under a microscope. Even as you transition to take other observations or maybe use some higher resolution methods, it might be important and interesting to continue your microscopy the way that you have for those 80 years to keep your long time series going and see how that environment is changing over time in a way that you can easily compare to this really important record that's been established for that site. As we talked about, it's cool that you can see the larger cells. That's also a weakness of this method, is that it's harder to see smaller cells. And a lot of the phytoplankton diversity is contained in those smaller size classes. So you might be missing a lot of diversity if you use a microscope. There's also some human error in identification. So I'm certainly not perfect at identifying phytoplankton taxonomy. I know there are people who have spent, thousands of hours more than I have, and they still make mistakes sometimes, right? And so that's just going to be a necessary part of any of this type of human classification method. Also, I mentioned here that you can't revisit your identifications. So on Monday, you did this IFCB exercise, and you had all of these images that you were able to cut out and look at and discuss and then put in order. And you were able to do that so many times because these images were saved. And so if you have a light microscopy image, maybe you could capture, especially in some of the newer higher resolution microscopes, you can connect them to the computer, you can take an image, you can revisit the site, the field that you were viewing, but you might not necessarily be able to have it on a cell by cell basis, and you will only have that picture that you took. You can preserve your samples, but that can sometimes change how the cell structure looks, or maybe change the amount of detritus in your sample, make it more difficult to see some of the cells. So you're a little bit limited in how often you can revisit your classification if you're worried you maybe made a mistake or you missed something major. And so I'm putting at the bottom of all of these cells the output from this method, because I think when we're thinking about what we're getting from an in-water method and what we might be comparing to a remote sensing method or an optical proxy, it's really important to remind ourselves of what exactly is being measured. So in this case, you have a volume of water, you have cells in that water, so you're counting cells per volume. And you can maybe make some estimates about sizes and convert that to biomass and maybe convert that to carbon. But for the most part, what you are identifying with a light microscope is cells, counting cells per volume in these different taxonomic categories. All right, we're gonna move on now to flow cytometry. Flow cytometry, again, you're taking a really small sample volume, around 1 milliliter. So if you think about, that's a really, really small amount of water. And the cells, the water can either be preserved and run later, or it can be run immediately on a flow cytometer. But what this is doing is it's counting and giving you both fluorescence and scattering information about individual cells. So the cells are directed down this flow cell that, like the IFCB, sees one cell at a time. It gets scattering and fluorescence information. And then you can plot that scattering and fluorescence information to show you these distinct communities of cells that you can identify based on their fluorescence and scattering properties. So flow cytometry is really, really useful for separating Synechococcus and Prochlorococcus. We talked about how Synechococcus has phycoerythrin that fluoresces a different range than chlorophyll, and that can be used in flow cytometry to separate those two major groups. If you don't have any other way to separate them, that's fantastic. You're getting these huge communities of cells that are really different from each other. You also can use stains or dyes to maybe give you some slightly different classifications or separations. So you have information about size, you have some information about pigments, and you might also have some information about specific structures within a cell that have been stained or dyed a certain way that then leads them to fluoresce differently as they go through a flow cytometer. If you have other information about your phytoplankton community, you can maybe get a tiny bit higher taxonomic resolution. So some people have looked at communities where there are a lot of cryptophytes. We talked about how cryptophytes have that unique pigment alloxanthin. And so if you have cryptophytes in high cell abundance and you know that they're in the water from other methods of detection, you might be able to separate cryptophytes also in your flow cytometry. But for the most part, we can just get different eukaryotic cell classes and then different cyanobacteria size classes. Weaknesses, it's really only looking at the smaller cell sizes. And in terms of taxonomic resolution, you're really only getting that for the cyanobacteria groups. You have a very, very small sample volume. So anytime that you're using a small sample volume for any of these methods, you're potentially missing the rarer cells in the environment, which is often the larger things in the environment. So for flow cytometry, since you're focusing on the smaller things anyway, your method is maybe going to be biased toward those smaller things, but you're also missing the larger, rarer things by having such a small volume. Flow cytometers also require calibration and they require really careful handling. And so thinking about, as oceanographers, we're going out to sea, we want to be often taking these high resolution measurements and getting information. Maybe you want it close to real time. Maybe it's really important on the cruise that you're on to know if there's Prochlorococcus in the water because somebody else wants to be able to take other measurements and use those measurements to do something, I don't know, with lipids or viruses or something beyond what you as an optical oceanographer are focused on. If you calibrated your flow cytometer on land and then you brought it on a ship and the ship is vibrating and it's rocking, your calibration might have changed. So that can be a real challenge for doing this at sea, which is a reason that people then might want to preserve their samples and take them home. But it also just requires, being really careful with your instrument, as a lot of these high resolution techniques do. Yeah. Yeah, that's a great question. How do you calibrate it? So you can get standards that will give you, either you can use cells like in a culture. So you have things that are a similar size and that are at a similar growth phase, or you can use these fluorescent beads that are similar to a lot of the other things that you use to calibrate other optical instruments. And so they have known scattering properties, fluorescent properties, sizes. And so when you see then where those fall within your instrument, then you have a better sense of where other communities of cells are falling relative to your standard. And so again, that output, so you have a volume, you have counted cells per volume, right? You also can estimate, because for Prochlorococcus and Synechococcus, that in terms of the cell shape, there's relatively little variability. They're pretty cylindrical. I heard somebody say over here when you were talking about different properties before, like they're as close as you can get to a perfect sphere. And that's pretty much true in the ocean, right? We don't have a lot of perfect sphere phytoplankton, but Prochlorococcus gets pretty close to that. So if you can make some estimates about your cell size, your cell shape, the amount of biomass in a cell of that size or shape, then you can have an estimate of the biomass per volume and, convert that to something like a carbon estimate per volume. All right, I won't spend as much time on this one because I feel like after looking at the wall over there, you all are pretty familiar with imaging in flow cytometry or quantitative imaging methods. I think a lot of people who use these methods have never done something like that. So that was a great exercise to be able to see what it's like to really be hands-on with those images. But in this method, again, you're using a pretty small sample volume, but it's bigger than the past two methods that we talked about. And in part for an instrument like the IFCB, it's going to depend on the sample time. So your sample volume will be collected and you'll know from the instrument how much volume was used. But if you have more cells you might sample slightly less volume in the same amount of time that you would go through fewer cells and a larger amount of water volume. So you'll get your small sample volume and then larger, in this case, nano to micro phytoplankton that are enumerated and imaged. You'll also collect that scattering and fluorescence information as part of your measurement. So for each cell image, you have the actual picture itself, and you can use that to talk about phytoplankton taxonomy, but you also have these other cell properties as they pass through the instrument. And so you know about the scattering and fluorescence properties. And so you can maybe use that information to tell you something about how those cells are similar or different to each other. Is this the same group of cells that you've identified as being in the same taxonomic class? Do those all have the same ratio of fluorescence to scattering, or are there things that are different? What does that mean about the phytoplankton community, right? There's a lot of potential there. So in my opinion, the strengths of this instrument, a huge one, is that it can operate autonomously. So you can have an imaging flow cytobot on something like a buoy or on a dock or a pier, and it can be operating without a person standing in the room and pressing the button and having each sample go. So if you're thinking about ways to monitor a system without having a human effort needing to be right in the room and collecting the sample and running it through the instrument, this is a pretty great way to do it. Obviously, you need to be monitoring your data quality and making sure that there aren't issues with the instrument, because as I'm sure anybody who's worked with this type of instrument can tell you, sometimes things happen and it can also be a little finicky. But it allows you to be sitting in your lab 30 miles away and pulling up your dashboard online and seeing what's in the water today, and maybe making a decision about if you want to come collect other samples, if you want to come take other measurements, if you want to take advantage of whatever is growing to come do an experiment, something like that. Also, the fact that we have these images, again, mentioned this a little bit with the microscopy, but you can come back and revisit your taxonomic classification based on new information that you get, based on higher quality information about the phytoplankton community that you get from other methods, maybe that you're deploying at the same time. You can just come and get all kinds of new information and reclassify and go through it iteratively if you're interested, because you always have those images saved as long as you have space for them on a server. In terms of weaknesses, it's only, again, biased a little bit toward those slightly smaller sizes of cells. So in the nano to micro range, the larger, rare things are just more rare in the environment. And so if you're only sampling 5 milliliters and there was only 1 in a liter, you're not going to catch that one cell. But we can understand that there's some uncertainty inherent in that method. And it also requires calibration and to some degree classification of your images. So you all did manual classification the other day when you had pages and pages of images and you're able to cut them out and personally stick them on the wall. But if you had, hours worth of data from a week of sampling, let's just start with a week, although people have a lot more data than that from the IFCB, you need to make sure that the different groups that you're identifying, you're going through and training your classifier. So you can have an automated way of sorting those images into different categories based on their features, but that requires a little bit of oversight as a human to make sure that the machine learning is doing it the way that you expect it should be doing it. In this case, the output, again, it's going to be per volume because you know your measured volume, you know the count, the number of cells, the type in each, the number in each taxonomic class. And you can also, again, get a biovolume estimate and convert that to carbon. All right. I'm moving on to molecular methods. I see a lot of people's eyes glazing over a tiny bit. So before we talk about molecular methods, which is a little bit challenging, everybody stand up for just 1 second. Just 1 second, you can do it. It's going to help. And just shake your body out really quickly. Stretch, like roll your eyes, roll your neck. You're doing great. Okay. All right. When you're ready, you can sit back down. So I said, I've kind of made a joke about this before. This is what used to lose me in terms of talking about detecting phytoplankton in water. I would hear molecular methods or DNA and I would zone out a little bit because it felt intimidating. And honestly, I felt like it wasn't going to be that useful to what I was interested in doing, which was using optics to look at phytoplankton communities. I have been converted and I am now a believer that these molecular methods are important and that they're going to continue to be important for providing us different information than other in-water methods for looking at phytoplankton communities. And so I think as optical oceanographers, it's important to at least engage a little bit with some of these methods and have a preliminary understanding so that when the molecular people start talking about their data sets, we can engage a tiny bit with them. You don't see one of these relative abundance plots and immediately like, oh man, all right, I'm just going to tune out for a minute, because that's exactly what I used to do. And we're going to try to get through it together. I also think that when the people in the molecular world hear all of us talk about optics, they feel the same way. So if we can just remember that and remember that we're all speaking different languages, but we can find a common ground, I think it could be really cool to have more people who feel really comfortable putting these tools together and using them to do more than they can do on their own. All right, so when I talk broadly about molecular methods, what we're talking about is using DNA from different types of phytoplankton to describe which phytoplankton are in the environment. In this case, because phytoplankton are so small and they have such small concentrations of DNA, we need to filter a lot more water to get at some of these questions. So if you think about in one cell in your body, you have DNA, in every cell in your body, you have DNA. In every phytoplankton cell, there's going to be DNA, but you only have one cell that's telling you something about one group. So we need a lot of water. We filter all that water, take our filters back to the lab, and then the next step is to extract the DNA. I'm going to talk a little bit more about this. And then take these barcode genes that we can amplify and sequence. So if you think about, you're at the grocery store, you're doing self-checkout and you're flashing the barcode on all of your different items. Each barcode is unique. That's what the barcode gene is referring to with DNA. So it's telling you whether it's this diatom or a member of the dinoflagellates or just a eukaryote. But that barcode is unique for each individual that you're sequencing, so it can tell you something about who's in the environment. So the strengths for this method, you can get to really high taxonomic resolution. For some groups that have been really well characterized, you can get down to the species level, which is insane to me. That's so exciting that you can use these little molecular tools and you can say, I know I have Phaeocystus antarctica in the water. That's really cool. You also can compare it to other methods pretty favorably in some cases, and we'll talk a bit about that. But for some broad groups, we can see that the relative amount of DNA compares well to the number of cells that have been counted under a microscope, or to the amount of pigments, and especially the amount of DNA and the amount of chlorophyll often scale pretty well together on broad scales. So we know that we're saying something about the phytoplankton community that compares well to other methods. In terms of the weaknesses, so I was talking about this barcode, you're scanning your barcode. You know, sometimes you like scan the barcode and the self-checkout thing is like, you need help. That's in this case, that can happen with DNA too, right? It doesn't identify the barcode. So the primers that you're using, which is how you tell these sequencing machines, like this is the part that you need to read, they don't always work for all organisms. So sometimes you can get this error and you might not be able to detect a certain organism that you're interested in because your method is falling short. I also put up this note that gene copies does not equal abundance. So what you're counting with these DNA methods is a barcode that's in each gene, but a phytoplankton might have multiple copies of that gene. So instead of having one gene in one cell, you might have 10 genes in that one cell. And then you're thinking, okay, I have 10 dinoflagellates here, but you actually only had one. And so that varies between different broad taxonomic groups. And we only know some of the limitations around that because people haven't been doing a great job of comparing across methods. A lot of people who are more comfortable using these molecular methods are taking them and saying, this is the truth, this is what's in the water. And so as we start to combine molecular methods with other methods, other people can say, wait, no, it's not. We don't have 80% dinoflagellates. I only counted 6 dinoflagellate cells in my 2,000 cells that I looked at. Right? So you need to have these combinations of methods to be able to understand some of the weaknesses and limitations. And I think with the molecular methods, we're a little bit behind on that, and we need to keep comparing methods to catch up. So then this is the last important thing about the method generally. Your output is relative sequence abundances. So for all the other methods we talked about, you're getting counts. You're having absolute, you're able to compare one sample to another sample. With the DNA methods, you have to look at everything as a fraction of the whole because the number of sequences that you have doesn't actually mean anything in the environment, which kind of stinks. It would be cool if we could count the DNA and then say that it is meaningful. We only can compare it as a relative abundance to say something meaningful in the environment. So I'm showing this example of the type of plots that you'll see with DNA data, whether you're looking at, different size classes or different types of phytoplankton, you're only able to look at it as a fraction of the total amount of DNA that you measured for it to make any sense. Luke. Yeah. Yeah, okay, so that was a great question. Is there any way to relate gene copies to cell abundance? I think that one way that you could try to do that would be to have cells of a certain group in culture, and you could use one method to count the number of cells and then another method to sequence the amount of DNA. And so people have done that with a limited number of phytoplankton groups because that's pretty difficult and time consuming to be able to both have your cells in culture, count them, and extract the DNA and sequence it, and then say, I have this many copies in this many cells. But also, that might be really different in the environment. We know that for a lot of phytoplankton traits, things are super different between culture and environment. And so you can say, all right, in this dinoflagellate that's been in culture for 12 years, we have this, gene copies per cell. That might be different across all the different taxa in the environment. So I think there are definitely people who have tried to do that. There are some papers that publish numbers for their study and say, within our cultures, we combined this amount of this one and this amount of that one, and this is what the genetic makeup looked like. I don't know of anyone who's really tried to do that in the environment in a quantitative way. The only other way that you could try to fix that is to find a barcode gene where you knew you only ever had one copy per phytoplankton cell. And so that's an active area of research, is people trying to find these other genes that they can use beyond the ones that have already been described and characterized, and for which we have these really detailed taxonomies. But that takes a huge amount of work because it's taken decades for people to relate these barcode sequences to a specific taxonomic group. All right, so everybody seems like they're not ready to die hearing about DNA so far. So we're going to talk about it a tiny bit more. Really broadly, thinking about molecular methods. This is giving an example where you have everything in the environment, but if we think about all the different types of phytoplankton, might as well be everything in the environment. So the first thing you need to do is collect your DNA. Then you're going to extract DNA from all your different organisms, so you can see all those different colors are represented in the test tube. The next thing that you need to do is amplify your gene of interest. So in this case, we want to measure this one gene. And if each phytoplankton only has one copy, or maybe only 10 copies, I don't know, that's still not that much to measure. So we want to be able to find that gene and then make multiple copies of it so that we can compare within the sample multiple copies of the gene and make sure that we're seeing the same thing. That's a step where a lot of problems can happen. So when we're talking about all these issues and these variations between method and this kind of user error, if you're assuming that they're amplifying evenly across the different types, there could be problems there. If you're assuming that within the amplification, it's the same as what you had in the environment, there could be issues there. So again, this is why we need to look at relative abundances, because if you have everything in your sample and then you multiply them all by 10, some could equal 10 and some could equal 12 and some could equal 25, right? So again, you're looking at that fraction within the group, but that fraction could change a lot based on some of these small problems that pop up. And I think they're still pretty poorly characterized for a lot of these different groups that we're interested in. So then once you have that gene amplified, that's when you do your sequencing step. So in that step, you have primers that you've attached to the start and end of the gene, and you're able to read the sequence that's between your two primers. So that sequence should be different for each organism. And again, you can see all the different colors that are corresponding with the different organisms. And in that case, we refer to that as the hypervariable region of the gene. So within these different phytoplankton genes that we're targeting, we know the hypervariable region within the gene. So we can design these primers to attach at the front and at the end, read what's in between. And then that part that you read is what comes out as your output, and that's what you associate with that specific sequence in terms of its taxonomy. So let's just talk about this really specifically for phytoplankton. These are not the only approaches, but these are the most common approaches that you'll find in a lot of different work being done right now, looking at phytoplankton ecology. So the 18S gene and the 16S gene are found in different groups of phytoplankton. As we talked about, different evolutionary paths, different ways that we got here. That means that the types of genes that different phytoplankton have can vary. So the 18S gene, super useful for eukaryotes. It's not in bacteria. So if you want to characterize your phytoplankton community, but you aren't as worried about the cyanobacteria fraction, you should use 18S. So you can take, you have your gene, you have your region of interest, you read your region of interest, your hypervariable region, and then you're able to assign those different regions to different phytoplankton taxonomy. We also have the 16S gene, which is found in the chloroplast. So it gets a little bit tricky for eukaryotes because some eukaryotes have chloroplasts that came from prokaryotic phytoplankton. So if you have cyanobacteria and 1 billion years ago, something else engulfed that cyanobacteria, but it kept the same gene, and you use 16S for eukaryotic DNA, you might get a really different story than if you used 18S because you're reading the chloroplast, but it maybe came from something other than the organism that it's still in now. So 16S is a way that you can get both the bacteria and the eukarya, but you might have some other complications. So with 18S, I said the big group you're leaving out is cyanobacteria. With 16S, unfortunately, the big group that we miss is dinoflagellates. And so that's a group that there's a lot of interest in detecting accurately in the environment. And it's really difficult to detect them because dinoflagellates are thieves. And over the course of time, they've stolen chloroplasts from tons of other organisms. So I'm going to show you a little diagram of that a bit later on, but keep in mind 16S, not for dinoflagellates. All right, this is our last DNA visualization like this. Again, I know a lot of people kind of zone out when they see this, but using these type of methods, when you have really high global sampling coverage, like in the Tara Oceans expedition, allows you to characterize the same organisms across different ecosystems using these standardized methods. So if you're using something like microscopy, you might have slight variations in how you're classifying things when you're in different places because the rest of your sample looks really different. But if you're using DNA methods, you're always going to be using the same primers, the same type of DNA extraction, and the same type of classification for your sequences. So if you're detecting one organism in the Southern Ocean and one organism in the Mediterranean, and it's the exact same sequence, that tells you something really cool about the phytoplankton that you're seeing across the whole globe having similar DNA to each other. And so in terms of our understanding of who is where and when, DNA can allow us to do these big global surveys and get a better sense of the distribution of different types of phytoplankton in different areas of the ocean, and then relating that to maybe the physics or the chemistry or other things that we can measure in the ocean environment. Yeah. Yes, that's a great question. So yeah, so for having the size component also come into the DNA, you need to think about your filtering. You need to be thoughtful about the sizes that you're measuring. So for Tara Oceans, they were extremely careful about separating phytoplankton sizes in their filters and then using the same DNA methods in the different sizes. And then what they did is look at those sizes separately here, but they've also had approaches where they've merged the data to look at them all together across the whole size spectrum. So if you're concerned that the size fraction isn't going to be well represented, if you're not filtering it in this size fractionated way, that's a really important way that you can introduce one more way of separating your phytoplankton community into a method like this that otherwise is just catching everything in these big bottles, filtering it all down onto the same filter. Okay, so the last slide I'm going to show you, now we're going to just quickly look at, I mentioned that these methods can sometimes compare well to other methods that we care about. There are a couple of cool examples recently from published papers in different coastal regions. And then this is one that I've been working on for the open ocean, comparing DNA at these metabarcoding methods for major phytoplankton groups to other methods. So in this case, we talked about fucoxanthin being the diatom biomarker, right? We expect to find more fucoxanthin when we have more diatoms. What do we actually find when we look at relative sequence abundances of diatoms in the environment? The answer is it can be really variable. So the one on the left is from my lab mate, Dylan Catlett, who looked at a huge time series in the Santa Barbara Channel across seasons, across different nutrient regimes, across different phytoplankton communities. So within this diatom sequence abundance, he has lots of different types of diatoms. And then we're measuring pigments the same way for all of those samples. And you don't always get a good relationship, but sometimes you get a pretty good relationship. So it really depends on the type of time of year that you're sampling and what's in the water, whether there's a good relationship between relative sequence abundances for diatoms and pigments. In the West Antarctic Peninsula, looking at the fraction of diatoms from a pigment-based method and the relative sequence abundances, for a lot of the year, again, you sometimes have really good relationship, and then there are outliers for that relationship. And for this data set, which is combined from the North Atlantic and North Pacific, fortunately, we had variability in the North Atlantic cruise in the relative fraction of diatoms, the amount of pigment. In the North Pacific cruise, we didn't have as much variability. So you can see there's kind of this big cluster of data all over there at these lower relative sequence abundances. But over the whole the course of these two data sets, we did see pretty good agreement between diatom sequences, diatom biomarker pigment. So more and better comparisons like this need to happen if we're going to be able to compare between these different methods. And the more samples that are being taken to use this molecular data and say interesting things about the ocean without comparing them to other methods that might be more relevant for optics, the more difficult it's going to be to reconcile those methods. So I'm a big proponent of encouraging people to try to take these samples in the same places and times. And if there are people that you know at the institutions where you're working or on cruises that you're going on who are trying to decide what types of samples to take and they can only take one, that's obviously they're going to decide on the best one for their method. But if they can take 2 or 3, you might want to think about how these methods can then compare to each other. So that can be a really cool way to start to look at limitations in the data set and what they can each tell us about phytoplankton communities. All right, now I'm super excited because we're getting to my favorite phytoplankton method. Ivana sent this beautiful photo of phytoplankton pigments from cultures in these clear bottles. So these are phytoplankton, their pigments are being expressed. You can see that when you have these monospecific cultures and they have really different pigments, you can get really different beautiful colors across your bottles. So what we're going to talk about in this context is using those pigments for their own sake, and then also using those pigments in the context of optics to try to separate different phytoplankton groups. I know people also might have some preconceived notions coming in about how well or not well this can work. And so if you have opinions about it, I would love to talk about it. So please feel free to express them as we're talking. We can have a little bit more of a discussion about if this actually is a way that works to separate phytoplankton groups or not. So giving this kind of broad method overview, for phytoplankton pigments, we also need a large sample of water. You can't use that 1 milliliter sample for flow cytometry. Pigments are in relatively low concentration. You have to filter a lot of water to be able to measure your pigments. You take your filter back to the lab and then use a method called high performance liquid chromatography, or HPLC, to look at how the amount and types of pigments varies between your sample. And as with all of these methods that we've talked about, a good HPLC system requires really good calibration. So again, you need to get standards to know the amount and type of pigments that you're measuring at different places along this chromatic graph that I'm showing on the top. And so which of these peaks correlates to different pigment types, and then how high the peak is correlates to how much pigment there is in your sample. And if you don't have a system that's well calibrated with using these different standards and knowing where you expect to find the peaks, you might not be able to separate pigments at high enough resolution. So if you look back at historical phytoplankton pigment data sets, oftentimes you'll see that there are only certain pigments measured, or maybe 2 pigments are grouped together because one of those peaks was actually pretty fuzzy and they couldn't tell which was which. But when money and time are put into developing a really high quality calibrated system, then you can get really good, consistent, precise, accurate data. So a huge service that the NASA Ocean Biology Processing Group provides for the ocean optics community is having this extremely well calibrated, very, very precise HPLC system that they run to be able to get this high quality data that then has good standards for across the community. So thinking about that data quality is really important in this method because it can be a little bit tricky to use. So a major strength of HPLC is that you're measuring phytoplankton. Anything that fits on your filter is going to be measured. So you're getting things across size classes from pico to nano to micro. There's also this really nice direct link to absorption and ocean color. So in terms of thinking about your goals or your methods for detecting phytoplankton, if you're interested in relating your phytoplankton community composition back to an optical measurement or remote sensing measurement at some point, pigments might be a pretty desirable thing for you to pursue, because we know that pigments absorb differently at different places across the visible spectrum. And so we can use some of those differences to try to relate them to differences in the optical signatures that we're looking at. The major weakness, we will talk more about this as I go through, and you've already had a little bit of a sneak peek at this. Many major pigments are shared between many major phytoplankton groups. So there's a lot of difficulty in separating more than the amount of phytoplankton groups that a pigment data set will let you separate. And I'll talk about that more as we go on. Your output is pigment concentrations. So you have an amount per volume, an amount of pigment, which is usually measured in either micrograms or milligrams per liter or meter cubed. All right, so when we think about phytoplankton pigments for the sake of taxonomy, we've talked about these really broad taxonomic groups. Typically, these are the associated marker pigment or pigments that you might see associated with those groups. So as I said before, fucoxanthin, the diatom biomarker pigment. That's great. We have a biomarker pigment that's in diatoms. We can use it to detect them. But is it only in diatoms? No, we already know that it's not, right? And this is going to repeat for other pigments throughout these different groups of phytoplankton. And you also saw how broad the diversity is in some of these small groups. So if I say chlorophytes or green algae in one ecosystem, that could be something entirely different than in another ecosystem, because all we know is that it contains chlorophyll B, and we know there's a huge amount of diversity within the chlorophytes. So now we have to bring this back again to evolution, how phytoplankton got into the ocean, how they formed all these different branches across the tree of life. That can tell us something about phytoplankton pigment composition. So when we think about moving from, and there are some time stamps on here, moving from these early freshwater cyanobacteria that are then becoming single-celled organisms with nuclei and moving into the ocean, there's this primary endosymbiosis. So that's what creates a nucleus in a eukaryotic cell, differentiating from prokaryotes to eukaryotes. And then the secondary endosymbiosis is what gets really complicated for pigments. So as phytoplankton evolved over the last billion years, some of them were consuming via these different feeding strategies other phytoplankton and ended up having really similar pigment composition across really different taxonomic groups. So on the left side of the image, you can see some of these major groups that we've talked about. We have the red algae with the red arrows, the green algae with the green arrow, and then the cyanobacteria with these kind of darker black or navy, I can't really tell, arrows. But you can see that there's going to be some similarity in how they all got there. Now, if we look at that in much higher resolution, you can see that as this red algal lineage evolves over time, there are going to be branches coming off where different phytoplankton pigments end up in similar taxa and vice versa. So what do I mean by that? In one case, we have the example of fucoxanthin that we've already talked about that ends up in taxa across the red algae. But we also have this example of the dinoflagellates being sneaky little thieves again and stealing chloroplasts from groups across the rest of the tree of life. So you have dinoflagellates with chlorophyll B that stole from the chlorophytes. You have dinoflagellates with fucoxanthin that stole from the diatoms. You have dinoflagellates that even have cryptophyte pigments. So yes, alloxanthin is unique to cryptophytes, except for that one dinoflagellate that ate it a long time ago that has been found in a very limited range of places. So most of the time we know that's not what we're detecting. But if you're in the North Sea, you might need to be careful because that might be the dinoflagellate that ate a cryptophyte. It might not be a cryptophyte if you're using pigments. So this is kind of a mess, right? And it also gets back to that challenge for 16S sequencing, where if you're looking just at the chloroplast, you're only detecting what's in the chloroplast. So you might not actually be detecting the rest of the organism if it stole its chloroplast from somebody else a long time ago. So we can kind of distill these challenges for pigment-based phytoplankton community composition into 3 broad categories as far as I see it. So this one we just talked about, many major phytoplankton groups share pigments either due to their evolution or due to their feeding strategy. There are a lot of different phytoplankton groups that have been observed in culture. If you put them in a co-culture with Prochlorococcus, what do? They consume that Prochlorococcus and they enslave its chloroplasts and use it for energy. So all these different mixotrophs are across the red algae. There's a mixotrophic chlorophyte. There's all kinds of crazy phytoplankton feeding strategies going on if they're in the same environment. The next major challenge is that within the same broad taxonomic group, you might have really different pigment composition. So in this case, I want to use the example of the Primnesiophytes. We talked about them having really huge differences in cell size and morphology. As you look at that broad taxonomic group, that also means really big differences in their pigments. So if we think about, they have these 3 major pigments. They have fucoxanthin, just like the diatoms, and then they have hexfuco and buttfuco. So those 2 accessory pigments, in addition to fucoxanthin, can be in different proportions, in different compositions. You might have one, but not the other. You might have only fucoxanthin. You might have 2 of them at the same time, but one is produced in much higher relative concentrations because of the cell growth environment, which leads me to this last point, that the growth environment for a phytoplankton cell can also impact the amount and type of phytoplankton pigments. So they might either upregulate or downregulate the production and expression of pigments based on the environment that they're growing in. And those words then relate us, unfortunately, back to the DNA world, where we're thinking about pigments as being produced on this cellular molecular level. Mix down to a deeper depth, and all of a sudden you have way less light, so you need to upregulate your pigment production. Then you have a ton of pigment, but then you get mixed back up to the surface, and that's where I scoop you up in my bottle. So I think that you just had a ton of pigment, and there's a ton of phytoplankton cells here, but it's just one cell that came from a really deep depth. So it was creating a lot more phytoplankton pigment. So within all this craziness, how do we use this information? We know these limitations to maximize the utility of pigments without taking these conclusions about phytoplankton community composition from pigments out of context. And I'm putting that another way because you are all optical oceanographers. Some of you might use pigments in the future. How are you going to use your pigment measurements responsibly? And I know that's kind of a big word to use in terms of like, okay, what responsibility do I have? But I think it's a pretty big deal because if you're interpreting the results of your pigment-based phytoplankton community, that could have implications for other broader applications. So if you're saying, I know that there's paradenin, the dinoflagellate biomarker pigment in this environment, and somebody uses that information to drive a policy decision and say, okay, the scientist told me there's paradenin, that means there's a dinoflagellate, that means there's a harmful algal bloom. I would want to be pretty sure that I was giving a range of uncertainty or maybe using another method before somebody was basing a big decision on something as variable as pigments. So how do we use it responsibly? I'm going to provide one possible route forward, which is that the pigment data to some degree will tell you its limitations. So you can use statistical approaches to see how many phytoplankton groups you can get based on pigments to separate from one another. And the big conclusion that I'll tell you right up front is that this is going to depend on your data set. It's going to depend on your environment and who was there and what was happening. So in this analysis, we looked at a global pigment data set distributed across latitudes and longitudes, all different seasons, all different phytoplankton communities. And we found that we could only statistically separate these 4 really broad pigment-based phytoplankton groups. The notable thing that I want to point out here is that people often use the paradenin to separate dinoflagellates. In this analysis, dinoflagellates did not separate from diatoms. So in this data set, on this scale, I would never suggest that somebody take paradenin concentrations to mean that there are dinoflagellates there because they're not separate enough from the rest of the phytoplankton in the red algae. You can say that you have diatoms and dinoflagellates, but you're not detecting just dinoflagellates in this data set. However, If you look at other more specific data sets, you might get a different answer. So this is now looking on a local scale. The top one is from the Santa Barbara Channel. The bottom one is from Palmer Station in the West Antarctic Peninsula. You might remember those were the two places where I also showed comparing DNA and phytoplankton pigments. So we have a lot of different types of measurements in these environments. They're both these long-term coastal observatories in really different ecosystems. So we get more and different phytoplankton groups separating from pigments than we did on global scales, because we have different phytoplankton communities occurring over the seasonal cycle. And we're going to be able to separate those groups differently using pigments because the pigments are expressed in different concentrations and compositions. So it's really important that for your question, for your ecosystem, for your data set, you do some of these types of analyses with your pigment data to tell you the limitations, what you can and can't say. And so my appeal to you is to please use your pigment data responsibly. If you perform an analysis saying that you got 16 phytoplankton groups using pigments and somebody sends it to me to review, I'm going to do a big, deep, sad sigh, because I don't believe that you did find 16 statistically different pigment-based phytoplankton groups, even though some methods that are out there might tell you that you did. So if you want to talk more about this, I love talking about this. I'm happy to chat about it whenever. Send me an email, talk to me at the class today, anytime. Use your pigment data responsibly, please, because it's a powerful tool and it does link to optics, as we're about to talk about, but we're not going to be able to use it to detect every individual species in the environment. Okay, we're doing great. We're getting to our last major method, optical proxies. So specifically, I'm going to talk today about absorption by phytoplankton and their pigments. There are also other methods to relate optics to phytoplankton taxonomy. I did not have time today to go through every single one, so you're going to get a little slice of this. And if you're interested in these methods or other ones, I encourage you to definitely keep looking into that. Those could be cool things to maybe develop for a project if that's something that you want to do while you're here at the class. So you already learned a lot about particulate absorption and filter pad absorption by phytoplankton. You got to practice those techniques already this week. You're familiar, I assume, at this point with what this type of absorption spectrum looks like. We also can look at phytoplankton pigment absorption. So you take the pigments that are in, that you filter onto a filter pad, and you can extract them into a solvent. So something that those phytoplankton are going to, the pigments themselves are going to be extracted out of the phytoplankton cells and dissolve into this solvent. And then you can measure the absorption of the solvent. So you know that you're looking at those pigments, but in this kind of background environment. So first, let's talk about how we can use phytoplankton absorption to relate it back to the pigments. You've already seen this figure where pigments have different absorption peaks. And so if you think about the variability in the phytoplankton absorption spectrum compared to other absorbing constituents in the ocean, we can take advantage of that high variability and these different pigment absorption peaks to try and separate a phytoplankton absorption spectrum into different phytoplankton pigment concentrations and composition. So a really cool example that I want to walk through for a moment is this one from Allie Chase, who will be here later this month to talk a little bit about phytoplankton and remote sensing. Allie did this great analysis using not just phytoplankton absorption, but particulate absorption measured with the ACS. And she looked at the data set from Tara Oceans. So there's a ton of variability in the magnitude and shape of the particulate absorption coefficient. What she found is that she could create a model summing together a dissolved component, a particulate component, and then these different Gaussian functions that are representative of the different phytoplankton absorption peaks, and find different broad phytoplankton absorption concentrations derived from the sum of these Gaussian functions. So I'm showing, this is the major equation in her paper that looks really overwhelming when you first see it on the page, but it actually is just breaking down the contribution of each of these little bumps to this overall shape, which is something that I think you've all been thinking a lot about already as you've been comparing the different spectral shapes of your natural seawater or your culture. You already know that there are differences in pigment absorption and phytoplankton absorption. So you can do something just like this with your pigment absorption, your phytoplankton absorption, You're summing up all those little Gaussian function components to try and match your original measured spectrum. And then what Allie did, because she had paired HPLC pigment concentrations, was developed relationships between the magnitude of absorption for these peaks and the actual measured phytoplankton pigment concentrations. And so the result of that is that you can, from an ACS absorption spectrum, get an estimate of pigment concentrations for major pigments like chlorophyll B, which we know is in chlorophytes, chlorophyll C, which is in a bunch of the red algal classes, and the summed either photosynthetic or photoprotective pigments that can tell you something about the status of the phytoplankton community. So this is a way to go from absorption back to pigments. We also can go from pigments to absorption. We have measured phytoplankton pigment spectra from individual, I was talking about standardizing your HPLC system. We can measure individual pigments from these standards in solvent and then see what the pigment absorption spectrum looks like. We can measure our phytoplankton pigments and then multiply them by their individual absorption spectra and sum those together to get a pigment absorption spectrum if we don't have a way to measure that pigment absorption in solvent on its own. And so one important thing to think about as we're doing all of this, extracted pigments, pigments in cells. I know Meg mentioned a little bit the other day about phytoplankton absorption in the pigment absorption in the cell. And she talked about how what you're actually looking at is the absorption of a pigment in a chloroplast in a cell, right? So this idea of pigment packaging came up. And that comes up again when we're thinking about using optics to relate it back to phytoplankton community composition. This idea of the package effect, so your pigments being packaged in a chloroplast in a cell, we're going to see some variability in the chlorophyll-specific absorption coefficient for different sizes of phytoplankton and with different chlorophyll concentrations. So this diagram that Colin put together, I think illustrates it really nicely, that when you have a larger cell with more pigment in that cell, the path length through the cell is really going to change compared to a different sized cell or a cell with less pigment. So you have this effect, and this is, I will, for Ivana's sake, say a very young paper that shows this example of at different cell sizes, your normalized chlorophyll absorption shape is really going to change because the way that the pigment is structured in that cell and the way that you can measure the absorptions in that cell is going to differ. So if you see, it's going from this, the line with 7 right here is the largest cell. The line with 1 at the bottom is, we don't really have a 0 micron, I guess, but the smallest one that you can think of. And so your shape, especially in these regions where phytoplankton pigments are absorbing, is going to really change, and the magnitude is also going to change. So I just put together, talking about going from absorption to pigments, pigments to absorption, a really quick comparison from actual data that we have in the environment. So comparing one of those ACS AP spectra, this is just one from Tara Oceans, a surface sample in the oligotrophic Pacific. And then I also took the pigment concentrations that were measured at the same station at the same time and multiplied those pigment concentrations by these exact specific pigment absorption spectra, summed them all together to show you what that looks like when you're comparing it. So you're not comparing apples to apples, even though you think that you're thinking about, in both cases, pigment absorption in a cell relating to phytoplankton. You're getting really different answers, both because of what you're measuring, right? So you're measuring all particulate absorption for the ACS, but also in terms of how those measurements were created and standardized. So in solvent right here, so you're not measuring it in the cell or in water, you have this shifted absorption peak. So if you're going to compare these types of measurements and maybe use something like absorption line height to compare to your HPLC measurement, you might have to shift the spectra and kind of like push it in the middle. And methods have been proposed for how to do that from people who have actually taken the time to do these really high resolution measurements in the same places and times across growth conditions, across seasons, across phytoplankton communities, to look at the extracted pigment absorption, the pigment absorption in the cell, the actual pigment concentrations. So all of these different relationships come into play when we're thinking about then the amount and types of phytoplankton that are contributing to these different signals. Okay, I'm going to put up a slide that might make everybody freak out now. So take a second with it. We've been talking a lot about methods. I was talking about units a little bit. Don't forget all the different units that we can be measuring phytoplankton in. This is a tiny bit overwhelming because oftentimes we're thinking about comparing one to the other, or we're thinking about using one to validate the other, or we only have relative sequence abundance, but we're using that to say something about carbon per volume, right? So Ali put this slide together for a lecture that she taught at the IOCCG summer optics course last year. And I liked it so much that I had to steal it for this lecture, because I think this to me is just distilling down this whole challenge of all our different in-water methods, all the different things that they're measuring within the phytoplankton cells, and all the different ways that those phytoplankton cells are varying from each other. And then if we want to standardize this, and we want to go out into the world and say, okay, we have our satellite image, this is what's in the water. Which one of these things from the in-water method are you using to validate it? And how does that alter the model that you're building for your satellite method, or the conclusions that you're drawing from your phytoplankton community that you're applying to a carbon export model? I think we need to really take all of these considerations into account when we're talking about community composition. All right, it's time to kind of shake things up again. So now that you are more familiar with common methods for describing phytoplankton community composition, we're going to practice. I have 3 quick case studies. You're pretending that in this case, you are a PI, a project manager, you're making the big decisions. You need to detect phytoplankton community composition. I'm going to tell you something about your system and your goals, and I want you each to decide what your ideal method would be for detecting phytoplankton community composition in this environment. So your first case study is a coastal observatory, maybe something like the Bowdoin Coastal Studies Center, I don't know, but you have easy access to get to your observatory with a boat. You can go out and sample. You also are really concerned about a seasonal harmful algal bloom that forms by dinoflagellates. This is an active fishery in your region. So people are going to be fishing, collecting shellfish. They need to have a timely warning if there's a harmful algal bloom to make sure that people aren't consuming infected shellfish or fish. So in this case, what would your ideal method be for detecting phytoplankton community composition for this observatory? You can just think about it. You don't have to write it down. You can if you want to. But if you feel comfortable sharing, let's talk about it. Okay, use an IFCB. Why would you use an IFCB? Awesome. Okay, so instant, you get the data. It can be sent out to other people and distributed. Are there any drawbacks to using the IFCB? Totally. Right. So you get mostly larger cells, small volume, but continuous sampling. Awesome. Okay. Does anybody either agree or disagree? Yeah. Okay. Right. Yeah. Absolutely. Right. So it's spatially limited. You're getting your point samples. And so you might need a way to relate it to remote sensing to get a bigger picture of your whole watershed to be able to alert people in other areas than where your IFCB is measuring. Awesome. These are great answers. That's exactly along the lines of what I was thinking in terms of agree, disagree, methods together, methods on their own. All right. So case study 2, in this case, we have an open ocean site with monthly time series sampling. You want to characterize all phytoplankton and you're interested in the seasonal succession. So you want everything and you want to know when and where. And wouldn't this be nice? Budget is no issue. So don't worry about restricting yourselves. What methods would you use and why? Okay, all of them because you have the money. What happens if your methods disagree? What are you going to do? Learn something. All right. Perfect. Yeah, I wish somebody had this existing time series and we could use all the methods and we could get this great comparison. There are things that get close to it out there, but it would be really cool to be able to have that as kind of a model ecosystem. All right, so this is our last case study, and it's maybe slightly political, but let's pretend you have a month-long research cruise to a dynamic high latitude site and you want to understand the links between phytoplankton taxonomy and carbon export. You're also tasked with developing remote sensing data products. What type of methods are you going to use to characterize your phytoplankton community and why? Yeah. Pigments, okay. Why are you going to use pigments? Awesome. Closely translatable to remote sensing. Yeah. Would anybody disagree with that? Would you like to add a method? Which one would you want to add? Yeah.
Optical properties of that status.
Okay.
Yeah, that.
Okay. All right. So adding some optical properties. Awesome. Anything else? Yeah.
I'd like to see that cloud.
Okay. Thinking about clouds.
Want something. Feeling like it's
Totally. Okay. So you need to make sure you have in-water methods to be able to, when you can't see the surface ocean from satellites, have your in-water measurements. Cool. Yeah. Cool. Okay. Yeah. Timing throughout the day. So when would you try to time your sampling on this cruise? Mhm. Yeah, definitely. Right. So thinking about that variability throughout the day. Absolutely. And so what about linking it to carbon export? Luke, were you raising your hand or stretching? Okay. Okay. Why do you propose another way of measuring phytoplankton taxonomy? Yeah. Absolutely, right. So if you're only using pigments, but you're trying to develop these important relationships, if you have some ambiguity in that taxonomic relationship, you might also want another method to supplement the pigments to tell you the uncertainty on those pigments, even if you're going to be able to link them to remote sensing. Yeah.
Need more information about what the distance of particles in the water, as well as how the particles interacting and sinking. So I think
Yeah.
in water sediment type things, taking samples and analyzing them or observing them in the lab, how the molecules are sinking or forming aggregates of sinking. So we need to know about particles.
Yeah, right. Know about the particles. Yeah. Totally, right. So timing your sampling during the day, so you have your optical measurements with your phytoplankton measurements, with your particle measurements, so you have them all at the same time. All right, you all just designed a mini exports. Great job. You're doing great. Okay, this is pretty much the end of my presentation. I want to leave you with the results of this survey that Ivana led as part of the PACE project, asking PACE data users what they're hoping to get out of PACE in terms of describing phytoplankton taxonomy. So she gave a few answers for people to choose from, but also made an other category so people could choose their own, or they could say specifically what they wanted. And these are the results that we got from over 100 responses. Yeah, so a lot of people participated with this survey, and this is what we have. So my question to you, how realistic do you think these goals are for PACE? And can we even do this in situ from what you've learned today? And you don't have to answer it now. I just want you to kind of keep it in the back of your mind as you go through the rest of the class. You learn more about remote sensing methods. We're facing a big challenge in terms of integrating in-water data and remote sensing data, but it's really exciting because we have so many cool methods to choose from. We're going to have such different data from PACE than we've ever had before for global hyperspectral remote sensing. So I think it's a pretty cool time to be interested in detecting phytoplankton in water and from space, but you've learned a lot today about how that might be really challenging. Okay, that's all I have. If you have questions, I'm happy to answer them now, but you've asked great ones. So.
This is part of the paper that Sacha contributed some Holly contributes on this. Currently sitting in Dr. Riddell's table, so if somebody can shoot him an email. I hope it's gonna answer a couple of questions that you have, so if you can give you the draft before we submit it. Hopefully, like in a week, when Jeremy comes, so you can give you draft. These beautiful reviews that, you know, review that's not just talked about, it also takes it a little really sense thing and things like that. It's a good thing whenever Jeremy fixes the things that he has a problem with.
Okay, we're about 5 minutes from lunch. So we're gonna eat lunch and then we're gonna be back here right at 1 o'clock. And we wanna, it's gonna talk something about fluorescence, and we're going to do some fluorescence. Okay? All right, let's think something.
Thank you. You guys are great. Thank you.
Ask Sacha open questions during lunch.
Yes.
That's time to eat.
Perfect. What have one said? Charlotte.